the next study, this is also a crucial study, IVPT selectivity, and the recommendation is the parallel assessment of reference test and alter formulation. The question was related to the minimum number of donors and replicate. I would take, like, whatever we discussed for the sensitivity study, 3 would be a good number, but 4 to 6 donor would be ideal, using 4 to replicate. So I believe that's the recommendation, or that's the suggestion from the panel. If there are difference in opinion, please provide your feedback. And then another thing that that I'm hearing from the panel, that once you identified, just have enough effort during the method development, and once you have all the parameter established, it would be a good practice or best practice that don't change any parameter going into the validation study or pilot study followed by the pivotal studies. And what are the considerations for the alter formulations? So I know we are at 3 o'clock. Dr. Rand, would you like to make some quick comment on what kind of consideration they can use for the alter formulation design?
Well, look for something here. I would say that you have reason to believe mechanistically, based upon the way the formulation behaves, that it should give you a dramatically different bioavailability. So you can really show well differentiated. Because hopefully in the pilot study, I'm not sure if you want to keep it down at 3 donors. Here, I think you want to get more information out of the pilot study. 3 donors, 4 donors, I think that makes sense for the sensitivity studies during method development. When you get into the pilot study now with, it's really kind of supporting your validation, I'm not sure that 3 donors is really enough. I think you want at least 4 donors here with 4 replicates, and potentially more, 4 to 6 even, because that might give you better quality information for predicting what you need for your pivotal study. And in terms of that altered formulation, if you're working with a cream, you might want to look at the altered one being a gel or an ointment or something like that.
Yeah. On the altered, we can also look at, the whole discussion we just had on the sensitivity. That should also give you some insights into the altered design. If the design is extremely dose sensitive or duration sensitive, you have to pick your altered formulation based on that. Typically, viscosity modulators or chemical penetration enhancers in the formulation can change the profiles drastically. So instead of going away from the formulation, sometimes you can look within the formulation also how drastically it can change, particularly looking at the sensitivity data.
Are we now confusing what the intent of the altered formulation is by calling it altered? The implication is you start with your base formulation and you make a change to it versus changing the whole base formulation, comparing a gel to the cream or an ointment to the cream. Is it just a different formulation with the same drug at the same concentration, or does it need to be a truly altered base formulation?
I would start with the same base and then alter it. And if you're unable to, perhaps we can move to a different dosage form.
I think that's a good idea. The challenge here is that if you choose a relatively similar formulation, so typically I would say choose something that has the same strength. But if you're choosing a relatively similar formulation and you don't see a difference, how do you interpret those results? Is it because, well, that formulation may just not have had a very different bioavailability? And I think that's one of the advantages with choosing a substantially different formulation, is that you'd expect that mechanistically to have a very different flux profile. And you want to be able to see, using your developed method, whether you can actually find for a product that you really expect to have a different profile, am I seeing a different profile?
I think, Sam, if you're looking for ways to streamline this process, going with a different dosage form is easier. Then you wouldn't necessarily have that risk of altering the formulation. I think you have a better chance of success seeing differences between dosage forms.
Exactly, and I think that's a great way to go. Is you really want to be able to show that under conditions where you really expect to see something different, that you do see something different. Otherwise, in those incremental changes, it may be difficult to interpret the results. So, just my opinion, and I'd be interested in yours.
No, principally, I agree with you on that. Again, as you know, formulation development takes a lot. suddenly you're giving a new formulation to be developed, which perhaps is not optimized. Is it stable? Is it separating? Is it appropriate for the altered formulation study?
Sure. So the intention here is not to create the burden of creating a new product just for this.
Yeah, exactly.
In all of the studies that we've done, we've just used marketed products for
If there
that
is
current
available,
formulation. Right.
if it's available 100%. My only concern was that suddenly you're now throwing another composition, which perhaps isn't even, discriminatory to begin with.
Okay, so we are at 3:05 mark. Thank you for your feedback. I'm just going to quickly run through the slide. The data analysis, we will be, I know we don't have enough time, so we will be talking in the next session. This is, I think we all know this is about reference product variability, and we always, refer this one as a aiming the moving target. So, I think we don't have enough time to discuss about that one, but this is a critical element when we talk about the topical drug products. And then my last slide is about designing the pivotal study, which will, because we don't have enough time, we will be talking in the next session. So with that, my sincere thanks goes to all these people. And last, but not the least, I would like to thank all our audience and panelists for a nice discussion. Thank you, and looking forward for the next session. Thank you, everyone.